Thank you for the introduction and thank you for having me here. Today, I'll be giving you a little bit of an overview of the program that we have set up at NCATS using engineered 3D lung tissues as assay platforms for drug discovery. Just to give you context, so at NCATS, for about 4 years ago, we established the NCATS 3D Tissue Bioprinting Lab as a multidisciplinary group of bioengineers, assay development scientists, and drug discovery experts to biofabricate and validate microphysiological systems and implement their use in the drug discovery and development pipeline. It does take a village to take these tissues, to develop, validate them, and use them for drug discovery. As the name sort of indicates, we started historically using bioprinting methods, but in the last few years, we've been expanding to other MPS platforms, including spheroids and tissue chips. And today I'm gonna give you some examples of the work that we're doing on this regard. Probably most of the work that we're doing is in collaboration with technology providers and disease experts, because one of the fundamental aspects when we start developing a tissue is what do we need to include in the tissue, and that it's really with the help of clinicians and researchers that tell us, well, I think you need to have these cells. And first we, like most of everyone is doing, first we develop the normal tissue, and then once we have a validated tissue, to the normal, then we introduce some sort of perturbation to create a disease model. We have a lot of emphasis on high throughput screening in our work. So most of the tissues that we're developing are in multi-well plate platforms that are compatible with lab automation found in HTS labs. And at the end of the day, really, if we can, and we use clinical approved compounds to validate, pharmacologically validate the model. So during this process, we do spend a lot of time validating the tissues, and by that means assessing that we have the right cell markers and the right maturation. Do we capture the right clinical biomarkers that we want to see in these tissues? And there's been a lot of talk in by previous speakers and this morning and debate discussion about like how you assess that these tissues are physiologically relevant. And it really depends on the questions that you're trying to ask, what disease. And the previous speaker gave a perfect example of that. And because we are very much HTS focused and screening focused, we're very interested in developing quantitative functional endpoints that we can use for screening. And again, I mentioned the pharmacological validation, and also something that came up this morning in terms of operationalization for HTS, the scaling up process, the assessing the robustness, the variation from well to well, the variation from plate to plate. So we can really use these platforms as drug screening platforms. At the end of the day, our goal is to implement these platforms in the drug discovery and development pipeline. So trying to make or help make better go, no go decisions when compounds are selected during the process. So a lot of the work that we're doing, as you will see, is establish fit for purpose. We have a lot of different technologies. How do you choose the right technology for the questions that you're trying to answer? How do you establish the context of use of each one of these models? What physiological complexity do you need? And what is the clinical predictability? We say we want to make these models because we think they're more clinically predictive. And the first question that you get is like, well, show me that they are indeed more predictive. So how do you establish that, right? So today I'm going to be talking about a platform of engineered 3D tissue models that we've been putting together in the last few years. Two words that I like a lot are modular and versatile. It takes a lot of work to create one of these tissues. And once you have the normal, let's call it the normal tissue, you want to try to leverage it for as many diseases as you can, whether it's cancer, for lung, whether it's cancers, whether it's infectious diseases or toxicity applications. So having a process to build these models and then being able to, in a plug and play way, to use them for different diseases is really something that is very powerful, and we try to do. So for the lung, and this is a general approach that we use in the lab, modular in terms of diseases, but modular in terms of physiological complexity. And that helps us sort of answer some of the questions in terms of exploring the context to use and exploring the fit for purpose. And I'm not going to go through some of the basics of the lung, but for the lung, we've been leveraging sort of existing commercially available tissue models of the different areas of the lung, whether it's the tracheobronchial, small airway, or alveolar. And also we are developing our own platform. So we have used sort of microfluidic platforms that are available. And really, our choices were based on a lot of it on throughput. As you can see, and there's a lot of different long MPS platforms. How do you choose? So in our case, we choose the Metas organograft plate because it gave us the throughput in terms of developing some of the models, and because we were able to leverage some of the protocols that we were developing for the more transwell models. So the models that I'm going to be talking today are sort of commercially available, sort of long epithelial transwell models. And then some of the vascularized lung tissue models that we are developing in-house, and some of the work that we've done with perfused vascularized lung epithelial organ-on-a-chip models. For each one of these models, essentially go through the sort of parallel physiological characterization, whether it's with histology, whether it's with microscopy. And lately, we've been using also single-cell sequencing in terms of assessing the different cell types that we have in the tissue. At the same time, these are very low throughput type of assays. So we use functional HCS measurements with the goal to really being able to implement screens, whether it's cell viability, whether it's measuring a tear, whether it's looking at cytokine secretion, and we leverage a lot high content imaging as well. So for the lung, we started using the commercially available. In our case, we use the MATEC transwell models that have been mentioned before. These are essentially epithelial models. So we use them because they were readily available. They were in a transwell, so they're sort of amenable to using instrumentation that we use for high throughput screening. So they come ready to use. You mature them in the ALI for 3 weeks. These are from primary epithelial cells, whether it's alveolar or from the tracheobronchial. And then we use these models particularly for as a sort of quick way to get into viral infection studies. So that was at the beginning of the COVID-19 epidemic. So we wanted to assess, can we use these models for studying viral infection or respiratory viral infections? So we use them, and then we apply different assays, whether it's direct measuring of viral infection using antigen staining and microscopy, looking at secreted virus. We also measuring multiplex cytokines. And then, as I mentioned before, we started doing validation using single-cell sequencing and as well as tier measurements. This has been out in a preprint and now has been accepted. So I'm just going to go quickly over the data, some of the basic data. So what we did was really systematically test the tracheobronchial tissues versus the alveolar ALI tissue models, in this case from MATEC again. And we assess infectivity. So in this particular slide, oops. I'm showing this is the MOX, so the blue is DAPI and the green in the middle is for the virus antigen. So this is the uninfected. So the top one is the tracheobronchial, the bottom one is alveolar. So you can see that these two are two strains of influenza, and this is one of the original strains of SARS-2. So you can see the differences with the different viruses in terms of time. So this is time after infection. So some are early, some are later, which is what was seen for SARS-2. And again, there's the differences between the tracheobronchial and the alveolar tissues that have been mentioned before in the in the previous stroke. So these are, I would say, simple, differentiated lung epithelial models, but they seem to recapitulate quite well some of the clinical features of the SARS-2 virus and influenza. So for this application, it seemed to be a good model. We also measure cytokines, and I'm not going to show all the data, it's in the paper, but one of the cytokines that clinically appeared to be that relevant for COVID-19 was IP10. So here I'm showing the data with IP10, and again, what I'm showing is for the tracheobronchial tissue, and then this is the alveolar, and you can see the differences between the influenza infection in terms of secretion of the IP10 cytokine, very quick, versus the SARS-2, which is takes longer time. There's a lag. There's less of it, and then the differences between airway and alveolar much higher in the alveolar for SARS-2 and also for the influenza. So you can tell the differences, and you can explore the differences between viruses and between tissues, and that sort of in agreement with the data that was shown by the previous speaker. Again, we did single cell sequencing, and I must say we're still digesting the data. It's a lot of data, but essentially we did it for the tracheobronchial, like non-infected, infected with influenza, non-infected, infected with the SARS-2, and then with the alveolar tissue, again, MOC, influenza, MOC, and SARS. And you can start seeing the differences again within the virus. We were able to detect all the relevant cells, but then we were interested in seeing the changes with each virus and try to correlate that with clinical responses. Can we sort of try to see if we can, in a way, use these tissues to predict clinical responses from these transcriptomics or single-cell transcriptomics signatures? And there's genes that are common and a lot of genes that are not common. So it's a lot of very interesting data that, again, we're still digesting, but sort of hoping that we can then go to the clinical data and do associations with these data in the tissues and see how closer we are in terms of predictability. We work with Dr. Sarah Cherry at Japan, and she had a platform for screening compounds in 2D. That was a high content imaging using CALU3 cells that were infected. And then they were using high content to look at viral antigens. So they did a screen of several libraries. And the idea here was to, again, can we use these ALI models to help prioritize the compounds before going into animal models? The whole concept of can we use these tissues to help make better selections? So this was published, but it was actually very useful. And here's some of the data for the SARS-2. You can see the infection here. This is the MOC infection. This is influenza. MOC infected, and then you can test compounds. And all these compounds actually were active in 2D, let's call it 2D. And you can see that some of them really confirm in the 3 models, but many of them didn't. So we think that these are good models if you want to prioritize compounds for putting into animal models. The limitation with these commercially available models is that what you get what you get, right? So if you want to start exploring additional physiological complexity, they're very limited. And in addition, they're like lower throughput. So we actually spend time trying to develop, we are developing 96-well plate trans-well models. And this is some of the data that we have generated with finding the right markers, the good tier values. And then the other thing that we're doing is we had developed protocols to include vascularized beds in the tissue. So we do have a transcriptomic data indicating that the transcriptomic signatures of endothelial cells in 2D or in monolayers are very different from what you see in sort of forming capillaries. So we developed protocols, publish this data. So I'm going to go quickly. So we mix endothelial cells, fibroblasts, parasites. We print them at the bottom of a transwell, and then after a week, we see nice angiogenesis and vascularigenesis. So we applied this protocol. We knew we could make the the long epithelial, could we make the vascularized bed under the long epithelial? So it's work in progress, but essentially we're taking a modular approach that was mentioned before. We take some protocols to make the vasculature and then protocols to make the epithelial. Can we put them together? And this is actually working quite well. So I'm going to go on the donut structure in a second, but essentially this we're doing in a 96 strands well. This is the bioprinted vascularized bed. And again, this is to show you the reproducibility of the protocol. And you can see this is, this particular model, and I'll go into it in a second, is for cancer. But if you don't add the small airway, you get this donut where you can see the vasculature forming. You can then add the small airway epithelial cells, and you do have an effect on the vasculature, angiogenesis and vasculogenesis. And this particular model was for cancer. So we added the cancer cells, and you can see the formation of the small tumors in the model. So again, showing you that we can develop these vascularized beds in a transwell format. And the reason to do a donut is it helps us with the quantitation. So we can easily create or use high content imaging to create mask and look at angiogenesis. So it's very important for us to be able to quantitate phenotypes if we want to use these models for screening. And in this particular case, it was a cancer model. So again, we can also look at the cancers. We can see whether the tumors are within the vasculature or somewhere else in the model. And that way we can, again, have a disease model, a quantitative assay for screening for this particular disease model. This model didn't have a perfused vasculature. It had a vasculature, it was in a transwell, but it didn't have a perfused vasculature. So we recently published this model. This is the one where we use the organoplate graft from me method. Essentially, it's a 3-channel chip. The outside is liquid. That's where we load the endothelial cells. And in the middle, there's a hydrogel where we add the mixture of the endothelial cells, parasites, and fibroblasts to make a vasculature. The beauty of these plates that we like is not only that it has throughput, but it has an opening here where we can put the long epithelial cells and create the air-liquid interface on top of the vascularized bed. And the protocol is actually very similar. It took a lot of work to optimize, but the bottom line is at the end, you can have a nice epithelial layer with very decent tier values, and you can, with the sort of modular way, you can investigate what happens if I don't have the alveolar cells, and really, if I have alveolar but not vasculature, and really you can start investigating the crosstalk between the cells and how that affects the function of the tissue. So we were able to make the epithelial layer in the ALI, and then we were also able to demonstrate that we could make a vascularized bed, and here are the markers with the microvasculature and the network, that it was perfusible. And we used a 70 kD dextran to actually show that we could fuse from one channel into the other channel. And we're still working on it, but again, I mentioned that we care about throughput. So we don't want to show just the one well that works here is the data from some recent experiments on one of these 64 plates, and it's actually looking very good. We have some of the wells that we're still working on, but overall we can see, and this is the progression from the beginning until the ALI, I think it's day 14. And here is just a blow up of one of the wells. So you can see the endothelial cells in one channel, the other, and the vasculature. And this is the whole plate with a couple of wells here that I think last week they said that they're coming up with protocols that look very good now. So scaling, reproducibility, it's important for us. And how do you know that you get the same in every well? So you have to quantitate. So that's what we're working on before I go into the quantitation, just to show you that it's not only that you can form the vasculature in every well, but there's perfusible. And here is I'm showing the data, and as you can see, it's not perfect yet, but we're working on it. And sort of playing with the conditions that you have the right sort of diameter of the capillaries. So. We use these models as work in progress again to see infection. So it's not only about seeing infection, it's also how does infection affect the vasculature. Is there any effect, direct or indirect, on the integrity of the vasculature? And that's what we are doing lately. So we are quantitating. We're working with Gilles Carpenter in France to develop algorithms that allow us to quantitate the integrity of the vasculature, whether it's a sprouting, whether it's mesh area, whether what percentage of the vasculature is really perfusing. And as you can see, when you infect, in this case is with influenza, some of the parameters don't change, some do change. And that's the beauty of these systems. You can look at different parameters and see which one changed depending on the disease model that you're creating. This is it for me. I think it's a quick overview, and just to remind everyone that it's not only just making the model, but finding the right cells. And really, you generate a lot of data, in this case, high content data, that requires a lot of support in terms of IT. So it's a whole infrastructure that you have to create to be able to use these models for high content screening and implement them as a pipeline. And this is the group, and thank you, everyone.